Good afternoon, I'm Ginevra Begani, and I will present you my work of the last 3 years of PhD on X-ray phase contrast imaging applied on neurodegenerative diseases using the Hartmann wavefront sensor. This is the outline of my speech, so I will introduce you the main concept of phase contrast imaging and the main application on neurodegenerative diseases. And of course, I will introduce the main concept of the Hartmann wavefront sensor and how it can be used for phase imaging issues. So, as I said, our main applicative part will be on neurodegenerative diseases. That is a set of 600 diseases that involve the loss of neural and cognitive function inside the brain and lead to neural cell death. This is a picture of the areas of the brain that are involved into neurodegenerative disease, and we will focus on Alzheimer's disease and on amyotrophic lateral sclerosis. This is a chart that shows you the disability adjusted life year, so the percentage of healthy life loss due to the occurrence of a neurodegenerative disorders. This chart shows several diseases, and as you can see, the highest percentage is recorded for neurological disorders. There are many ways to image the brain and soft tissue in general. And on one end, we have a 2D imaging technique. For example, we have LIPS, that is that is a technique in optical microscopy that uses laser-induced background spectroscopy to visualize the sample and give maps of several elements inside the tissue. We can use histochemistry, so we can label specific cells inside this tissue, and we can dye them. And we have electron microscopy that uses an electron beam to visualize with high spatial resolution the tissue. On the other hand, we have 3D techniques that makes use of large machines. And for example, we have MRI, so magnetic resonance imaging, that use high magnetic fields to visualize the internal structure. Then we have PET, so positron emission tomography, that makes use of nuclear, is a combination of nuclear medicine and tomography. And then we have CT, so what we are going to use in my thesis, so computed tomography, that normally is performed with the X-rays. So what are the main features about the imaging technique that we show? So for 2D imaging, we saw that they are very high spatial resolution, but they have limited spatial coverage of the sample, since normally is needed to slice the sample into several sections. So we have a destructive sample preparation. On the other hand, we have the 3D imaging techniques that give us volumetric information, but nowadays they lack in spatial resolution. So a nice combination of the two techniques is what we propose to use is X-ray computed tomography. So we will use X-rays to visualize the sample in 3D and to give high spatial resolution inside the tissue. So we can introduce how tomography works. So it can be performed with many kinds of sources. So as you can see here, we have the source, the sample that is put on a rotational stage, and a detector. What we do is that we acquire many images of the sample, and while it rotates, these images are called 2D projection, and they are collected at several angles of illumination. Then we need a reconstruction algorithm to visualize the sample in 3D. And finally, we will have the final volume where each pixel will be called a voxel, so a 3-dimensional pixel, and we can visualize the sample in 3D. Now I will present you the main concept of phase contrast imaging and what brings more in respect to the classical techniques. So in soft tissue imaging, what is normally performed is that the detector is placed just after the sample, and so there is no space of propagation. And this is an image of the brain performed in absorption. In this kind of images, the attenuation, so the contrast is given by the attenuation of the tissue and is proportional to the Beer-Lambert law, where the incident energy, so the incident, where the intensity is recorded proportional to the linear absorption coefficient of the tissue and to the object thickness. This is the equation for one element, for only one single element. And the linear absorption coefficient is proportional to the atomic number. Since in soft tissue, the atomic number is very, very low, we have very low absorption contrast from soft tissue. So what we want to do, we want to increase the contrast in our images, and what we want to use is phase contrast imaging. So we will start introducing every object that can be explained, can be described by the index of refraction. The index of refraction has delta, so the real part that is proportional to the phase, and beta that is proportional to the absorption of the object. These delta and beta, they have the atomic factor F1 and F2. When we look at higher energy photons and at low zeta elements, such as biological tissue, we see that the ratio delta over beta scales as the square of the energy. So here you see a plot of the delta and beta for brain tissue. And you can see that the contribution of delta is that is the blue line, is much higher than the contribution of beta. So phase imaging will be predominant over amplitude imaging for biological samples. So this is an example of an image of the brain that is performed in absorption. So this is the setup where we put the detector just after the brain. In this case, we saw a brain, and we see that the rays, they start to deflect from the sample, but we don't have enough space to detect this deflection, and we lose this kind of information, and we only have absorption contrast. We lose this information because the deflection angles, when we are speaking about hard X-ray imaging, are very, very small, of the order of microradians. So to be able to visualize them, what we do is that we put the detector much farther, and we can appreciate the deflection introduced by the sample on the X-ray beam. And as you can see, the image of the brain becomes much more clearer, and we can appreciate much more structures inside. And in order to do so, the propagation needs to be meters long. And on the detector, what will happen is that some rays, they will superimpose on a certain number of pixels, while in other they will disappear. So the edges and the structure inside, they will appear nicer and more detailed inside the sample. What we need with free space propagation phase contrast is that we need a phase retrieval algorithm. So each image to obtain from absorption image to phase contrast image, we need to apply a phase retrieval algorithm. One of the most famous one is Begani algorithm. That's what we used, and that is proportional to the ratio between beta and delta. So between delta and beta, so between phase and absorption. In this kind of images, phase and absorption will be mixed, so we cannot retrieve separately the two signals. So what I'm going to show you now is my results on neurodegenerative diseases. So they obtained with free space phase contrast imaging. We will concentrate on the brain for animal models, especially mouse. We will concentrate on the cerebral cortex of the mouse and on the hippocampus that are normally areas involved in neurodegenerative diseases. So the first set of experiments I will show you, it was performed at the SRF synchrotron. We did 3D imaging of the brain. And in this case, the brain was affected by an animal model of Alzheimer's disease that mimicked the human disease. We consider 2 time points, so 12 and 20 months old mouse. And these are some projections of the tissue inside the brain while we slice inside the volume. These are some images from the experiment. On the left, you see the healthy mouse. In the middle, you see the 12 months old mouse. And on the right, you see the last time point, so the very old time point of the mouse. What we are going to focus, our analysis will be focused on the brain plaques that you see here as bright dots that are agglomerates of protein that forms due to the progression of Alzheimer's disease. And on the other hand, we will look at brain fibers that connect the electron signals inside the brain and that are formed of myelin. And normally what happens with neurodegeneration is that the myelin won't have the same structure while the disease progresses. So the first step was to quantify the fibers inside the brain. So this is the image analysis, the analysis we perform on the image. So we define an ROI. We project the maximum inside the tissue and inside the volume. We threshold with the intensity value, and then we apply the skeletonized plugin to measure the length of the fiber inside the volume. This is the result of the quantification of the fibers inside the brain. This is an histogram of the fiber length for the 12 and 20 months old mouse. As we found that in gray, for the old time point, we found that there's an increase in the number of shorter fibers. So there's a clear difference between the 2 time points of the disease. Also, we quantify, as I told you before, the plaques inside the brain. So we threshold, we apply a similar image analysis as the fibers. So we threshold the plaques inside the brain, and then we count them, and we define the volume of each of the plaques using a 3D object counter plugin. This is the result in 3D of the quantification and of the volume of each of the plaques for the 12 and 20 months old mouse. And here you see the histogram of the volume of the plaques for the two mouse, and we see a very big increase of the number of smaller plaques when the age of the mouse was increasing. So another example of application of free space tomography, free space first contrast, was obtained on amyotrophic lateral sclerosis. That is another kind of disease that involved the spinal cord of in this case of the animal model of the mouse. We consider a presymptomatic stage of the disease, a 60 days old and a 90 days old. And the measurements were performed at the beamline ID17 of SRF. What we analyzed in this case was the motor neurons that are located in the ventral horn of the spinal cord, and then control the voluntary muscle contraction inside the tissue. Then we also quantify the vascularization inside the spinal cord, since we have seen that the permeability of the vessels is influenced by the occurrence of neurological disorders. So this is the quantification we perform on motor neurons. You see here the 3 time points of the mouse. So here you see the healthy mouse, here you see presymptomatic, and here you see 90 days old mouse. In the green inset, you see a magnification of the motor neurons, so that you can see in the image as bright dots. And we can clearly see a net decrease in the number of motor neurons when the disease was progressing. On the upper part, we see a magnification of the interneurons. So that is another kind of neurons, and until the presymptomatic disease, they were still present inside, while they had a net decrease in the 90 days old. This is the quantification we perform of the green inset, so in the ventral horn, of the number of neurons, and we found a net loss in percentage inside in several time points. Also, we quantify the vascularization inside the volume as the number of branches of the vascularization per millimeter cube. So you see the same 3 time points that I told you before. We quantify in the posterior horn, that is the red inset inside the brain, and in the anterior horn, that is here in white. And we found a much longer, a much higher loss in the posterior horn of the number of branches of the vascularization with respect to the anterior horn. This is a 3D rendering of the spinal cord, where in yellow you see the neurons, and in red you see the vessels. On the left you see the healthy mouse. On the right you see the 60 days old mouse, and it's clear even from this 3D rendering the changement of the number of neurons and the vascularization inside the spinal cord. So what we learn until now, what are the limitations of the current phase contrast techniques, is that they mix phase and absorption imaging signal, so we cannot separate them. We cannot calculate the exact density and chemical composition of the tissue, and the total setup size, it needs it needs normally meters of propagation to obtain, so it cannot be used in lab for the moment. So what our solution is to use the Hartmann wavefront sensor for phase imaging. So we want to be quantitative in the density and the chemical composition in 2D and 3D thanks to tomography. And we can retrieve with a single acquisition absorption, deflection, and phase maps. And the total size of our system is up to 50 centimeters. Now I'm going to show you the main idea that's behind the Hartmann wavefront sensor, so how it works. So what we want to sample is the wavefront, and how we can represent the optical phenomena. There are 2 ways. There is ray optics, so we can consider the light as rays that will propagate as straight rays as soon as is refracted, diffracted, or absorbed by an optic, or we can consider wave optics, so in the sense that the light will be considered as a wave and as a ray. And with this kind of, with wave optics, we can understand the phenomena like interference and diffraction. So what we want to sample is the wavefront, that is the group of points in space where the wave has the same phase. And there were several methods that were used during the years to measure the incident wavefront of a wave. And what we are going to use now is the Hartmann wavefront sensor. So how the Hartmann wavefront sensor works? What we have a source and a Hall array and a detector that is placed after a certain distance. What the Hartmann plate will do is that we will sample the oncoming beam and will create a series of beamlets. This series of beamlets will create a reference image of our beam. To understand and to retrieve the wavefront of the incoming wave, what we need to know is the center of each of the beamlets that was created by the Hartmann mask. And we are going to look at several algorithms to look at the center of each of the beamlets. Then when we put a sample inside the beam, what will happen is that each of the beamlets will shift, and the shift of each of the beamlets will be proportional to the local slope of the wavefront. So the each shift in the x and y direction will be proportional to the first derivative of the phase, and we will use a reconstruction algorithm to retrieve intensity and, of course, absorption maps and phase maps. So as I told you before, we need to calculate the center of each of the beamlet. So we test several algorithms to find the center of each of the beamlet. The very first and classical one is the first moment centroid that used the intensity weighted algorithm. Then we can use the weighted first moment. They use the weighting function multiplied by the intensity of each of the beamlet. We can use a Gaussian 3-point fitting that it just fits a parabola inside each of the spots. And we have more advanced algorithm like iterative algorithm and 2D Gaussian fitting. So to understand better the parameters and how to optimize the Hartmann wavefront sensor for phase imaging, we implement a simulation tool. So there are several open questions that we want to address in this kind of systems. So the first one is, what is the effect of the bandwidth in our system? What is the effect of the source size, of the level of coherence of the source, and also the propagation between several optical elements, and the design itself of the Hartmann plate, so the shape of the aperture and all the parameters. So we implement a Python code to address all of these questions. What we use is to propagate from one element to the other is the classical Fresnel propagator configuration. So what we have is that the field after a certain distance z is just the Fourier transform of a complex field and a quadratic phase factor. So the propagated field can be rewritten in a simpler form in this way, where H is the propagator and F are the frequencies in the Fourier domain. What we want to look at, we were very interested in our simulation tool, is to look at the level of coherence and to control the level of coherence of the incident beam. So how we did it? We took a Gaussian beam inside the source and we propagate up to a plane after a certain distance. Then we moved our Gaussian, we multiply by a quadratic phase factor and we propagate again. And we did that several times up to arrive to the final source size of our system. So what is the logic of our loop is that we define the source, then we apply the Fresnel propagator for one optical element to the other, and then we sum only at the end the electric fields to have absorption and phase maps. We want to validate our definition of coherence inside our simulation tool. So we test on the diffraction pattern from a sharp edge. This is the two images that we had from the simulation. And as you can see here, on the left, you have the simulated curve in black, and on the right, you have the theoretical values reported for a sharp edge diffraction. And in black, you see the result of the simulation for the coherent illumination, where you can see all the oscillation due to the diffraction from the edge, while in the coherent illumination is reported in blue, all the diffraction are smoothed, and since the source size is larger, and all the signal will be averaged between the several sources inside the source. So there were many open questions in the design of the Hartmann sensor that we want to answer. So there will be the first part, we divided in propagation problems, so related to the energy, to the distances, to the shape of the aperture of the mask. And on the other one, we had the detection problems. So we want to look at the performance of the detection algorithms on theoretical spots. Then what we did is that we merged these two parts and we look at the performance both on simulated spot and experimental spots for the Hartmann. Now I will start from the propagation related problems, so from energy and propagation distances. So this is the model of a very simple Hartmann wavefront sensor where we have the mask, the mask in between the source and the detector. Then on the left, we simulate the image on the detector for a high coherence case, where you see the diffraction from each of the aperture of the Hartmann. While going to the right, we simulate lower coherence sources, and we see that the shape that each of the beamlet is changing due to the change of coherence of the incident beam. So for higher coherence, we have the occurrence of crosstalk within the holes, while for lower coherence, we have almost a Gaussian shape, since the beam is, when we go a very low coherence, is just the shape is convolved with the shape of the source. So our source was Gaussian, our beamlet, they became Gaussian. This is a plot of one single beamlet for the coherent in purple and coherent case in blue. Then we look at the effect of propagation between the Hartmann mask and the detector. So we simulate several distances from the mask to the detector, and we look at two energies. So the upper row is for 10 kV, and for the lower row is at 20 kV. So then we move the detector 1 centimeter. We look how the diffraction pattern of the holes was changing due to the propagation after the mask. And finally, we arrive up to 5 centimeters, where it's clear that for the lower energy, we see many oscillations due to the occurrence of crosstalk between holes, while changing the incident energy. So at 20 kV, we don't see the same, we don't have the same problem. So these kind of spots, they will be easier to detect since they are sharper, and it's easier for the detection algorithm. Now we will move to the detection part. So we will look at the results of our detection algorithm on theoretical spots. Our goal is to arrive to 0.1 microrad of sensitivity, since we want to sample deflection in the hard X-ray image, in the hard X-ray energies that are the deflection are very small. So we want to arrive to 1/100 of the pixel size. So this is the result on a theoretical Gaussian spot for basic algorithm. In blue, you see the performance of the first moment algorithm, and in green, you see the performance of the weighted first moment algorithm. On the left, we change the level of the threshold of each of the spots. So each of the spots was threshold up to a certain value, while on the right, we look at a lower threshold level of each of the spots. These are the performance of the algorithm changing the full width as maximum of the Gaussian. So on the left, we see that the weighted first moment, it performs always better than the first moment, while on the right, we see that it increases in accuracy when the threshold of the spot is taken at a lower level. But even if the weighted first moment is better, is a better algorithm, what we found is that it's not sufficient. It's not sufficient because it only takes 1/3, 1/4 of the pixel size. So it doesn't increase a lot the resolution that we want to achieve. So what we did is that we look at more advanced algorithm. So we look at iterative Gaussian algorithm, where the weight, each beamlet is weighted by a Gaussian beam, and its position is iteratively adjusted. So you see here that this is the result of the calculation of the centroid for one iteration, while on the direction of the arrow we increase the number of iterations, and we go to the real center, increasing the number of iterations. Here you see a plot of how well they found the theoretical spots with respect to the number of iterations and the computational time. Then finally, we test our algorithms on an experimental spot. This is an image that was acquired at the Electra synchrotron at the beamline CIRMEP. We used the Hartmann waveform sensor from Imaginoptic. This is a magnification the single spots, and we see that the one that performs better are the Gaussian 3-point fitting and the 2D fitting in the 2D fitting algorithm. This is another example of another sample that we image that is the mouse backbone. Here you see a magnification. This is a raw image where each of the intensity correspond to the intensity of a Hartmann spot. If you look at the center, you can see the attenuation due to the rib of the mouse. And this is a magnified image, a magnified raw image where each red dot represents a calculated center from our algorithm. It's important to know that in each of these images, we have 1 million of centers to find, and to perform a CT, we have 1,800 images in this case. So we have many spots to calculate, to reconstruct the image, and so we need to be accurate, and of course, we need to be very fast. So what we learn from our simulation results is that we want to achieve 1/100 of the pixel size, and the two algorithms that performs better were the fitting, so the 2D fitting, and the iterative methods that iterate the center of each of the weight. Now I will present you the results that we performed using the Hartmann wavefront sensor on face imaging. So in this experiment, we use a compact Hartmann sensor from Imagine Optique, and what we want to look is that the effect of propagation from the sample to the Hartmann sensor. So we took an image at the near field, so close to what we call the near field, so close to the sample, and then we moved the detectors further, and we acquired one image at what we called in this case propagated field. We had two test samples. One was a PMMA rod, and the other one was a capillary filled with water. And with the Hartmann sensor, we can reconstruct transmission and deflection maps. What we can see is that in the slope, so in the deflection image, we see the presence of an air bubble inside the sample, while in the transmission maps, it is completely vanished. So what we did is that we applied the simulation tool I showed you before to simulate with the same parameters of the experimental condition. And these are the results for the near field, and on the right you see for the propagated field. In red you see the line for the modeling, and in black you see the line for the experimental data. We found a very good accuracy in the near field, and also the jump in the propagated field due to the propagation after the sample is very clear and very well representative for water, both in theory and experimentally. Then we implement a new prototype for the Hartmann wavefront sensor. So we had a rotational stage for the sample, we had a very big Hartmann mask, and a flat panel detector. To understand why we need to perform face imaging, we test with a snail shell. The sample was a snail shell, and what we did, we did a tomography experiment with this kind of sample, and after that we reconstruct in 3D the absorption and the deflection imaging. In this case, it was clear that in the deflection image we can look at several details, so the presence of cracks of imperfection on the snail shell that were completely absent in the absorption reconstruction. So this is the kind of complementary information we want to achieve with phase contrast imaging. This is another example of a biological sample. This was a mouse backbone. On the left, you see the absorption reconstruction. On the right, you see the deflection. Here, you see a magnification of the internal trabecular structure of the bone, while on the right, on the deflection image, you can appreciate more the direction inside the tissue of the main components. At the same way, since we apply tomography, we can reconstruct in 3D both the absorption and the deflection. We can have complementary signals, and we can look at the organization of the samples in 3D and how the several structures are organized together. Finally, we applied our algorithm on a mouse backbone with a different algorithm. What we did is that this is an example of the volume that we reconstruct in deflection. So here you can see the bone and the ribs inside the spinal cord, all surrounding. This is soft tissue, and all the small lines that you can see are the vascularization inside the spinal cord. So we were very happy about these results, and thanks to tomography, we were also able to reconstruct in 3D and to see complementary images of the same sample. I will show you now the experimental results that we performed with synchrotron light and to optimize the Hartmann mask. So this experiment was very important, and what we did is that we retrieved density and chemical composition of known elements. We performed the experiment at the electrosynchrotron at the CIRME beamline. This is a picture of the setup. So what we did is that we filtered the incoming white beam. Then we put the sample, and we used the Hartmann wavefront sensor from ImagineOptic. So this is an optical scan of the sample that were a set of microspheres, where you can see here the chemical composition of each of the spheres and also the zeta numbers. So the density of each of these, so the electrical density of each of these microspheres were very similar, so it's very complicated to differentiate them. This is an example of the image that we perform with the Hartmann sensor. So we look at the upper row, you see a beam, a polychromatic beam centered at 16 kV. At the lower part, you see a polychromatic beam centered at 22. And you can see the transmission, the deflection in the two axes, and the phase reconstruction for each of them microsphere. This is the results that we had for the calculation of delta and beta. So we repeat the same experiment with the microspheres with polychromatic beam before, and now with the monochromatic beam. So these are the results for delta and beta for several elements, so silica, silical oxide, and aluminum oxide. If you can look here at the energy of 10 kV, we see that the value of delta are very similar from silicon oxide and silica, while for beta, we see that the values are very different. So looking at the value of beta and delta, we are able to discriminate the chemical and infer the chemical composition of a known material inside the sample. Finally, I will look at the conclusion of my work. So what we look is that we can implement a 3D phase imaging based with the Hartmann wavefront sensor. We developed a simulation tool to visualize the sample. We look at the experiments both with the laboratory and synchrotron sources. We retrieve for the first time density and chemical composition of known material. And on the other hand, we also study neurodegenerative diseases with the X-ray phase contrast in free space propagation. And we quantify in 3D morphological features inside the sample. What are the perspectives of our work? So we want to apply and extensively this system on neurodegenerative diseases. So we want to implement more the 3D modeling of simulation. Also, we want to investigate the more specific biological problems such as amyloid plaques in Alzheimer's. And we want to use with the other structures, with in other way, the Hartmann wavefront sensor for plasma detection or for study the stress inside the material. And of course, we will work also on the implementation of the hardware and software skill. I would like to thank all the collaborations that we had in our PhD, in my PhD. There's a lot of people, so I hope I didn't forget someone. And thank you very much.